Okay, this is part 2 of our muscle tissue lecture. So now we're going to get into the microscopic anatomy of skeletal muscle cells and be able to look at the physiological process that has to occur to result in what we call a muscle contraction. So as we'll find in other places in anatomy, anatomists have made up special words to describe parts of a muscle cell. And you'll notice some common zoological roots being used repeatedly when we talk about muscles and muscle cells. So for example, we refer to the sarcoplasm. And when you hear sarcoplasm, you probably think of cytoplasm. And that's really exactly what the sarcoplasm is. Sarco referring specifically to muscle cells. So sarcoplasm is just the cytoplasm that we find inside muscle cells. Similar in its consistency to the cytoplasm. But additionally, it's going to have some inclusions, which we talked about being a part of a cell. So you have cytoplasm, you have organelles, and then you have other things called inclusions. So we have myoglobin, for example. And when you hear myoglobin, you definitely see myo, which is another root meaning muscle. And then you hear globin, which may make you think of hemoglobin. Hemoglobin, of course, is the oxygen binding protein that we find in red blood cells. So it's a pigment, but it is a protein in terms of our macromolecules. And so we're going to find lots of myoglobin in our muscle cells because oxygen is going to be important for some of these physiological processes, certainly for what we call aerobic respiration, which is going to be how most of the ATP will be produced for muscle contraction. So we tend to have lots of myoglobin in our skeletal muscle cells, and then also something called glycosomes. And when you hear that word, remember, you should look for zoological roots that are familiar, but also listen for some context or some reference, something that the word makes you think of, and that can often bring you to an accurate kind of introductory knowledge of the word. So glyco makes us think of sugar. We've talked about glucose and glycogen, right? Forms of sugar. So glycosomes are going to store glycogen, which can be converted into glucose. And again, that is a source for ATP production. So these are important things that we have in our muscle cells. Myofibrils. So again, we see the root myo for muscle. And fibril sounds like fiber. Remember, a fiber is what we call a muscle cell. So a muscle fiber is a muscle cell. And then you have that ILS at the end of the word. And that is a diminutive. So a myofibril is going to be a smaller part within a muscle fiber. Okay, so you've got that zoological root there to kind of clue you into the fact that a myofibril is going to be a smaller thing than a muscle fiber. So there are thousands of myofibrils within a single muscle cell, which we call a muscle fiber. Myofibrils are specialized in cytoplasm, okay, other than the normal kinds of organelles, so sort of like an inclusion as well. And so your myofibrils are formed from myofilaments. Again, myo for muscle, and filaments are going to be in the categories of thick filaments and thin filaments. And our thick filaments are going to be called myosin. We'll talk more about those. And our thin filaments have 3 protein components to them. The one we hear about the most is actin. So actin and myosin are special filaments. These are proteins again, and I think we might have mentioned them when we talked about proteins. These have a lot to do with creating movement. And so that makes sense because we're talking about muscle cells, and we know that the function of muscle is to generate movement. So our myofibrils are going to be formed from these thick and thin filaments. And the arrangement of the thick and thin filaments creates a unit called a sarcomere. And so we can think of a sarcomere as the functional unit of skeletal muscle. This is what we're going to really look at very closely understand its arrangement, and understand how it, again, ultimately results at that muscle contraction at the gross anatomy level. So this is all microscopic anatomy, but ultimately it leads to that muscle contraction at the gross anatomy level that we can see and that we're more familiar with, and that we're going to talk about creating movements of larger body parts. Sarcoplasmic reticulum, abbreviated SR, should certainly make you think of the endoplasmic reticulum, because that's basically what it is. It's sort of a specialized version of the smooth ER that we talked about being present in other cells. And remember, what makes endoplasmic reticulum smooth versus rough is the presence of ribosomes. So rough endoplasmic reticulum had ribosomes on it. Remember, ribosomes are where protein synthesis occurs. Smooth endoplasmic reticulum does not have any ribosomes, and that is the case here. But the sarcoplasmic reticulum, again, sarco telling you that we're talking about muscle cells, is going to have the function of storing and regulating intracellular calcium. So it stores calcium within the muscle cell, and we're going to see why that is super important for muscle contraction to be able to occur. And it also regulates intracellular calcium. So intra is a zoological root meaning within. We don't want to confuse that with inter, which would mean between. So intracellular calcium is looking at how much calcium is within the cell, really how much is going to be released into the sarcoplasm to allow for certain steps to occur for muscle contraction. Okay, so sarcoplasmic reticulum stores calcium and then also regulates how much of it is released from this storage place into the sarcoplasm, where it can then act to facilitate the steps required for muscle contraction. And the sarcoplasmic reticulum is going to be all around the myofibrils within the muscle cell. The part of the sarcoplasmic reticulum, as we'll see, where we have the storage of calcium, is called the terminal cisternae, and we'll look at that again more carefully when we have a picture up. And then the last specialized part of a muscle cell we want to identify is called the sarcolemma, and sarco again for muscle. Now you might be familiar with the term plasmalemma, which is sometimes what the plasma membrane of a cell is referred to as. So the sarcolemma is basically the plasma membrane around a muscle cell. And it's a little bit unique because it has these T tubules as part of it. The T in T tubule is for transverse. And the T tubules are invaginations of the sarcolemma into the muscle fiber that are going to allow the nervous signal that comes down from the neuron to sort of move through all of the muscle cell, and that ultimately will cause that calcium to be released out of the sarcoplasmic reticulum into the sarcoplasm, where we said it can then act to facilitate the steps in muscle contraction. So the T tubules are extensions of the sarcolemma, or invaginations, because they kind of dive down into the muscle fibers, and that's going to allow the signal that comes from the neuron to activate the release of calcium. And this sort of arrangement is going to help to keep the myofibril kind of operating together with all the other ones, because remember we said that there are thousands of myofibrils within a muscle fiber. And so there's going to be a lot of activity going on among all the individual smaller parts to result in that muscle contraction at the gross anatomy level. So here's a picture of all of those parts that we just identified. And the myofibrils make up about 80% of the volume of the cell. So there's tons and tons and tons of them within a single cell. And so you see on the left there that you're looking at one muscle cell or one muscle fiber, and all of the tiny, tiny parts within it are the myofibrils. And then they're enlarging part of a myofibril to show you some of those specific parts that we identified. So notice in blue is your sarcoplasmic reticulum. And you can see how it really does sort of spread all along and all around each individual myofibro. And all of those are held together by the sarcolemma within that one muscle cell. And they're pointing out the T-tubules, which are running down kind of across the length of the myofibro. And they're pointing out what's called a triad. So a triad, and we hear 3, so we know we're expecting 3 components here in the triad. The triad is made out of 1 T-tubule and 2 terminal cisterns within the sarcoplasmic reticulum. So I used the word before, terminal cistern or cisternae, and those were kind of the sac-like parts that are going to store the calcium. And so 1 T-tubule with a terminal cistern on each side is referred to as a triad. And so they attach to each other by proteins, and they're going to be important for the release of calcium, as we said, into the sarcoplasmic reticulum, right? So the nerve signal comes down through the T-tubule and then stimulates the opening of calcium channels within the terminal cistern to get that calcium into the sarcoplasm, where it can take the next step in muscle contraction. If you look at the topmost component shown in the picture, topmost myofibril, you don't see the sarcoplasmic reticulum on it, so they've taken that off so that instead you can view the sarcomere. And so those lines that they're showing you, the I band, the A band, the H zone, that's all part of the sarcomere. And again, that's the functional unit of a muscle cell. That is what is ultimately going to result in the contraction. Okay, so remember when we looked at skeletal muscle under the microscope, we observed that it was striated, that there were visible lines or striations cutting across the length of those elongated cells. And one other type of muscle was also striated, if you remember, it was cardiac muscle. But we're focusing on skeletal muscle, which is striated. And the striations that are visible on the myofibrils are really that contractile part of the muscle fiber, really making up the sarcomere. So it's a repeating series of dark and light lines that you see under the microscope along each myofibril that were creating what we call these striations. And the dark bands are part of the A band, and the light bands are going to be part of the I band. So we'll look more at that arrangement in just a minute. But here's that picture of what we saw under the microscope, and we didn't see it quite this close up, quite this clearly. But now that they're enlarging it, we do see the dark and the light, making it look kind of striped or striated. And they're labeling the dark as the A bands and the light as the I bands. And we're going to look at that arrangement more carefully in just a minute. So as we said, the myofibrils are organized into functional units called sarcomeres. Again, there's your sarco word or root for muscles. And sarcomeres are formed from the bundles of myofilaments. So remember, myofilaments were thick and thin filaments that create the myofibril, and the myofibrils created the muscle fiber. So it's kind of important to keep that hierarchy clear in your mind. So thick filaments are made up of one kind of protein called myosin that we'll look at. Thin filaments made up of 3 proteins. Actin is the one we're most familiar with, but 2 others, tropomyosin and troponin, are also important. So we'll look at those. And then there is a third type of filament, an elastic filament, that kind of holds the other parts to the edges of the sarcomere, which are called the Z bands or Z lines or Z discs. And these elastic filaments are made up of a protein called titin. And titin is a really, really large protein. Okay, so here's that picture again showing you a couple of muscle fibers. So those are the muscle cells that we saw under the microscope, and you can see those striations. The nuclei kind of flattened and pushed to the edges. And then in B, they're showing you again, kind of putting it into context. So you're looking at one muscle cell surrounded by the sarcolemma, which is like the plasma membrane. And then you see within that one muscle cell, many myofibrils. And then they pull one out so that you can sort of see the sarcomeres on it. And notice along a single myofibril, there's many sarcomeres kind of lined up end to end to end to end. Okay. Also within that cell, you see some nuclei. Those are the purple things. And then they're also pointing out mitochondrion, which is the singular of mitochondria, as we say it. And if you remember what those organelles do, you'll understand why we need lots of them in skeletal muscle cells. Remember, mitochondria are the powerhouse of your cells. They're where ATP is produced through cellular respiration, which we'll look at in the next lecture. And so an active, busy cell like a skeletal muscle cell is going to need a lot of ATP. It's going to tend to have a lot of mitochondria to support all of its physiological kind of cellular activity. Okay, so looking at the detailed organization of the sarcomere on a myofibril. Remember, sarcomere translates into muscle segment when we break down its zoological roots. And you've got two full sarcomeres here and some parts of a sarcomere. I'm sorry, one full sarcomere and then a couple of parts on each end of it. And so one sarcomere runs from Z disc to Z disc. Sometimes you see those as Z lines, but here they're calling them Z discs. So those are sort of the margins of the sarcomere. Okay, so one sarcomere runs from one Z disc to the other Z disc. We also have two I bands, and the I bands are made out of thin filaments. And so the I bands are going to connect to the Z discs at each end, and they are made up only of thin filaments. So where the thin filament attaches to the Z disc or Z line, that is your I band. And then you have an A band, which is where overlapping thick and thin filaments are. So basically, it's the length of the thick filament, so the sort of darker red running at the center of that sarcomere, that is your A band, but parts of it on the edges are overlapping thin filaments as well. So the A band is where thick and thin filaments overlap. Within that A band, then we can point out the part at the center called the H zone, where there's only thick filament, so there's no overlapping, and you can see there I'm pointing that out at the top bracket, H zone. And then they identify what's called the M line, which is where the myosin fibers divide the H zone. So the M line is kind of right at the center of the A band, okay, where the thick filament, the myosin, is kind of divided into two parts. Okay, so you can look at these pictures and we're going to see it in a slightly different form where it might be easier to see the parts. So I like this bottom picture better where they make things a little bit clearer. They also show you those elastic filaments that are holding the thick filaments to the Z discs. So looking at the bottom picture again, let's run through the parts. So one sarcomere is Z disc to Z disc, so you can kind of more clearly see those two ends. And then you have the thin filaments that you can see connect to the Z disc and run almost all the way to the M line. And so those are the I band, the part where the thin filament is not overlapping the thick filament. And then you have the A band, which runs the whole length of the thick filament. So the thick filament is the kind of peachy colored one. The thin filament is the blue one. And so if you follow from one end to the other of that thick filament, that is the length of the A band. But some of that overlaps the blue, right? Overlaps the thin filament. Where there is no overlapping blue, right at the center of the thick filament, that is your H zone. Okay, so this is the sarcomere, and we're going to get even closer and look at exactly what happens between the thick filaments and the thin filaments to result in that muscle contraction. Okay, so we just described that top picture. Now we're going to zoom in, like I said, and we're going to look at the details of how the thick filament and the thin filaments are arranged. So on the left-hand side, you see the thick filaments. And thick filaments are made up of myosin molecules. Myosin is a big polypeptide chain, so we've been calling them proteins. It's a big polypeptide chain, and one thick filament might be formed from 300 myosin molecules. Okay, so thick filaments are made up of many, many, many myosin molecules. And the arrangement of the myosin molecule is that it has two heads, a hinge region connecting those heads to a tail region. Okay, so it's got two little heads connected at a hinge region to a tail. And you can see the tail is where the two elongated portions of the molecule kind of intertwine. And so we'll see that those heads have two sites on them. One is called an actin binding site. So that's where it's going to be able to connect to the thin filament. Remember, actin is one of the proteins in the thin filament. And then it has an ATP binding site where, of course, ATP is going to connect. And remember, that is our energy source that's going to allow this connection to occur and result in our muscle contraction. Okay, looking at the thin filament. Remember, the thin filament is formed from three proteins. Actin is one of them, and the actin is the part that kind of looks like the blue beads. And then you have troponin and tropomyosin, and these are actually regulatory proteins that are going to shift to expose the myosin binding site on the actin and allow for actin and myosin to join together. Okay, so we will go over these parts again as we look more carefully at how they interact. Okay. So here's where we get into the physiology of what's going on to result in muscle contraction. And there are four major steps. The first one are the events at the neuromuscular junction. So we'll talk all about that. You hear neuro, meaning nerve, muscular for the muscle cell. So this is where the neuron and the muscle cell meet, and they're going to have to communicate. Now, the neuron, of course, is using electrical energy through that action potential, which we'll learn more about later. But when it gets to the space between the neuron and the muscle cell, that electrical energy can't continue across the space. So it has to be converted into chemical energy. And that's what acetylcholine does. It's what we call a neurotransmitter. And we'll see how it's released into the synaptic cleft, the space between the nerve cell and the muscle cell, to carry the signal across to the sarcolemma, which, remember, is the plasma membrane of the muscle cell. So that's the first thing that has to happen. Next, we have muscle fiber excitation. And so remember that we said that muscle is excitatory and it can receive that electrical stimulus, and that will trigger changes within the muscle cell. So we'll talk about what's called the end plate potential, triggering the action potential, traveling through that outer part of the muscle cell. The third step is called excitation, contraction, coupling. So when the muscle fiber is excited, basically, that electrical stimulus will cause the contraction to occur, basically first with the release of calcium from the terminal cisterns into the cytosol. So review these words. We introduced these words at the beginning, and now we're going to need to use them kind of fluently as we talk about what's happening. So the terminal cistern was that storage sac that was part of the sarcoplasmic reticulum, and the cytosol, remember, is part of the sarcoplasm, part of the cytoplasm within the muscle cell. That calcium is going to be necessary to allow the actin and myosin to connect. And then the last step is called cross-bridge cycling. So the myofilaments, the thick and thin, will shift following a series of events that occur after the actin and myosin connect to each other. And then at the end of that movement, the myosin heads have to detach from the actin. So we'll look at all of these steps. We're going to start with the neuromuscular junction. And after we complete this step, there'll be a little check-in quiz, there'll be a little video, and that'll end this part of the lecture. And then you can take a break and we'll go to muscle fiber excitation in the next lecture. Okay, so the first step in our skeletal muscle contraction are the events at the neuromuscular junction. Okay, so again, neuro for neuron, which is the electrically conducting nerve cell, muscular for the muscle cell. And so as I was saying, there is a electrical signal coming down the neuron and it needs to get to the muscle cell. However, where the neuron meets the muscle cell, there is a space and the electrical signal can't cross that space. So we have to convert the electrical energy to chemical energy. And that is done through the use of a neurotransmitter. So acetylcholine, ACH, is the neurotransmitter that's going to be used in skeletal muscle contraction. And we'll see how it's going to be released into the synaptic cleft to move the signal to the sarcolemma. So when you look at this picture here, and we're going to look at this picture over and over and over again, that yellow bulb is called the axon terminal. It's one of the endings of the axon where it meets up with a muscle fiber, a cell. And inside it, there are what we call synaptic vesicles. So those little circles with the green dots inside of them are synaptic vesicles. And the green dots inside are the neurotransmitter, the acetylcholine. You see some mitochondria in there. We know what those do. And then you can see the synaptic cleft is labeled as the space between this axon terminal and the wavy margin that you see is the sarcolemma of the muscle cell. Okay. So identifying some of these parts as I was just describing. You see in the bottom left picture the motor neuron, motor because it's carrying information out of the central nervous system to a muscle to make something happen, to make the muscle contract. So that's motor innervation, and we'll talk more about that later. So you see the motor neuron branching into, and they're showing you 3 of those axon terminals. And a neuron can have many, many, many axon terminals. And so each axon terminal is going to a muscle cell. And then the second picture is the one we were just looking at, enlarging that connection between the neuron at the axon terminal and again that wavy margin of the sarcolemma of the muscle cell. So the axon terminal is one of multiple branches at the end of an axon that extends very close to the sarcolemma. It's not actually touching it. It doesn't touch it because there is that synaptic cleft, that space between the axon terminal and the sarcolemma. And so as I was describing, it's an electrical signal that's coming down the axon to the axon terminal. And it's going to activate what's called a voltage-gated channel. And so remember, channels were one of those kinds of proteins, transmembrane proteins that we find in the plasma membrane. And a voltage-gated channel is going to respond to basically electricity as we think of it. Voltage is essentially the electrical potential energy, which is formed from the separation of charged particles. So positive and negative charges, right? Polarity is going to create our membrane potential. So we'll look at that a little bit more carefully later. But basically, it's that voltage gated channel that is going to be triggered from the action potential that's coming down the neuron to the axon terminal. And this will ultimately cause the contraction in the skeletal muscle cell. So we'll talk about two different voltage gated channels associated with the neuromuscular junction. That action potential, the term I just used a minute ago, is describing a large change in membrane potential that spreads over a long distance. So remember, membrane potential means we have positive and negative charges separated on opposite sides of the plasma membrane. So let's say a positive charge inside the cell and a more negative charge outside the cell. That would be establishing membrane potential. And when there is a significant change in that membrane potential, that separation of charges that travels a long distance over a cell membrane, that's called an action potential. So that's what came down the axon initially, and that is what we want to trigger across the sarcolemma of the muscle cell. But in between, we need to translate that electrical energy into chemical energy to get it to cross the synaptic cleft. All cells have a resting membrane potential, and then what we do is we change it through changing the permeability of the membrane, oftentimes through these channels, and that will create a change to the membrane potential, which translates into kind of electrical signals. Okay, so the signal is going to be converted from electrical, coming down to the axon terminal, to chemical as it crosses the synaptic cleft, and then it will be converted back to electrical to create another action potential along the sarcolemma. Okay, same picture. So the synaptic cleft is that space we've been describing, a small gap between the axon and the sarcolemma that we got to get that electrical impulse message to cross. And we do that with the neurotransmitter, which is a chemical messenger. The synaptic cleft is filled with kind of a gelatinous extracellular material. It has glycoproteins in it, for example, and the neurotransmitter is going to move across that space to get to the sarcolemma. So the neurotransmitter that we use with skeletal muscle is called acetylcholine, and it's a chemical messenger that is stored inside vesicles, right, which are those little bubbles that we see inside the axon terminal. The neurotransmitter is stimulated to be released into the synaptic cleft through the influx of calcium into the axon terminal. And the voltage gated channels are going to be what open up to allow the calcium to come in. So we can talk about voltage gated channels, which operate with that kind of electricity, and chemical channels which operate with neurotransmitters, for example. So for the calcium to come into the axon terminal, it requires a voltage gated channel. And then that calcium causes those synaptic vesicles that contain the neurotransmitter to bind to the axon terminal membrane and spit out the neurotransmitter into the synaptic cleft. Okay. And we've talked about this spitting out process before. That cellular mechanism that spits out something from a cell, remember, is called exocytosis. So exocytosis is what will get the neurotransmitter out of the synaptic vesicles in the axon terminal into the synaptic cleft. So exocytosis puts the neurotransmitter into the synaptic cleft. But then it has to move across that space to get to the receptors on the sarcolemma. And you can see the receptors are those little blue things in the picture on that wavy muscle margin. And so the cellular mechanism that's going to move a molecule from an area of high concentration to an area of low concentration, you know, is diffusion. So diffusion will move the neurotransmitter across the synaptic cleft because there's a lot of it where it's first released from the synaptic vesicles into the cleft, and there's not a lot of it on the other side. So that's high concentration to low concentration. The difference in concentration is called the concentration gradient. And so the neurotransmitter will diffuse across the synaptic cleft and reach those receptors that you can see in the picture on the sarcolemma, on the muscle cell. Those receptors on the sarcolemma are chemically gated channels. They aren't triggered by an electrical impulse, by changes in membrane potential. They're triggered by the attachment of the chemical, the acetylcholine neurotransmitter, to them. So on the sarcolemma, the outside of the muscle cell, the neurotransmitter acetylcholine will bind to these chemically gated channels, and that causes them to open, allowing sodium into the muscle cell and potassium out of the muscle cell. And see, we're moving positive charged ions, and that's going to change membrane potential. Remember, we said membrane potential is the differential in charges on opposite sides of a membrane. So we'll look at this in a little bit more detail, but basically the neurotransmitter crosses the synaptic cleft through diffusion, binds to these chemically gated channel receptors on the sarcolemma, which causes them to open and allow sodium into the muscle cell and potassium out of the muscle cell. This creates a small change in membrane potential, which is called an end plate potential. So we used the term action potential earlier. That's a big change, travels great distances. An end plate potential is a small change traveling a short distance. And if you look at the picture, they show you that purple arrow, and we've labeled that the end plate potential. And it's really only going to have to go as far as that next little pair of receptors that you see when you follow the wavy part of the sarcolemma over to the right. So it goes a short distance, which is why it's an end plate potential. Okay. And that neurotransmitter, acetylcholine, will be broken down by something called acetylcholinesterase, which is in the synaptic cleft, and that's going to break down the acetylcholine and end the response. So if there was a constant unlimited supply of neurotransmitter, then there would be a constant end plate potential happening, and that would not be good. We wouldn't want a muscle cell to be continuously without end stimulated. So acetylcholinesterase is going to break down acetylcholine into acetic acid and choline molecules. So that's an example of a decomposition, right? And then that will stop the end plate potential at the moment. I know this is a lot to take in, but the more you look at the picture, talk yourself through what's happening, watch the videos that you're going to watch, it'll become really easy for you to go through all of these steps. Trust me. Because we're going to do it one more time together, okay? Recognize that this is a neuromuscular junction, and there's one neuromuscular junction per cell, but each neuromuscular junction has many, many axon terminals. So the top part is the neuromuscular junction, and it includes, all of those axon terminals that are reaching multiple muscles. cells. Okay, so back to our picture of the neuromuscular junction. So that big yellow bulb is the axon terminal, and that was part of the motor neuron that brought the signal from the central nervous system to this particular muscle cell within whatever muscle we are ultimately contracting. And that action potential, which is coming down along to the axon terminal, is going to trigger the voltage-gated calcium channels on the wall of the axon terminal there. And that's going to allow calcium to enter the axon terminal. Calcium inside the axon terminal is going to bind to the synaptic vesicles, the little bubbles that contain the neurotransmitter, which looks like the green dot. So the calcium causes those synaptic vesicles to fuse to the edge of the axon terminal, and using exocytosis, spit out the neurotransmitter into the synaptic cleft. The synaptic cleft is that slight space between the axon terminal and the skeletal muscle's sarcolemma. So exocytosis puts the neurotransmitter into the synaptic cleft, and then diffusion moves the neurotransmitter towards the sarcolemma. Remember, diffusion is moving from high concentration to low concentration, so down your concentration gradient. And that is a passive mechanism. We'll talk more about that, but it does not require any ATP to occur. So the neurotransmitter, acetylcholine, will diffuse across the synaptic cleft, reaching the receptors on the sarcolemma. And the sarcolemma kind of folds up, as you see there, near the axon terminal, and that folding increases surface area for more receptors to increase the response. And we sort of talked about that way back when we looked at microvilli, how those extensions from the apical surface of an epithelial cell increased surface area. We've talked about the many bony projections on a vertebral arch, for example, increasing surface area. So here that junctional folds of the sarcolemma increase surface area for receptors and for that response. So the acetylcholine, the neurotransmitter, will diffuse across the synaptic cleft and bind to these chemical gated channels on the sarcolemma, and that causes a change in membrane potential. It allows sodium into the muscle fiber and potassium out of the muscle fiber, and as we'll see, those are both positive ions, but they move in a 3:2 ratio, which is why there's a change to membrane potential. And then that will create the end plate potential, the small change in membrane potential that will carry the signal on along the sarcolemma. Okay, so we're going to do a little check-in, see how you did with some of these introductory concepts at the neuromuscular junction. Okay, so take a minute to come up with the answers to these questions. You can pause the PowerPoint if you want to give yourself some time to think about it. I'm going to go on to the answers slide next and go over the answers with you. So pause it now if you want to take a few more minutes to think of the answers to these questions. Okay, so the organelle in a muscle cell that stores calcium is the B sarcoplasmic reticulum. Remember that it's a modified smooth endoplasmic reticulum for a muscle cell for the purpose of storing calcium, and we'll see why calcium is necessary for muscle contraction in the next part of the lecture. The other terms are also parts of muscle cells, but not the one that stores the calcium. Right? Myoglobin stores oxygen, glycosome stores glucose, and the sarcoplasm is just the plasma membrane around the muscle cell. Second question, the sarcomere is formed from A, thin filaments, B, thick filaments, C, elastic filaments. The answer was D, all of these. And the arrangement of those filaments created that sarcomere with the Z discs and the I band and the H zone. Question 3. At the neuromuscular junction, when the neurotransmitter crosses the synaptic cleft and reaches the receptor, which is on the sarcolemma, is B, chemical gated. So remember, it's being triggered by the neurotransmitter, the acetylcholine. So that's a chemical response. It's not voltage gated. It's not triggered by changes to membrane potential. We haven't talked about mechanogated or mechanical receptors are going to be respond to physical changes in shape. So it was definitely chemical gated because that neurotransmitter is a chemical. What causes the release of neurotransmitter from the synaptic vesicles? That was the introduction of calcium into the axon terminal. That calcium was released due to a voltage-gated channel from the action potential coming down the neuron. And then we saw that the calcium bound to the synaptic vesicles and caused them to fuse to the margin of the axon terminal to release the neurotransmitter. What cellular process moves the neurotransmitter across the synaptic cleft? So once exocytosis puts the neurotransmitter into the synaptic cleft, then it was diffusion that moved it across the cleft to the sarcolemma. Diffusion is a passive mechanism, meaning it does not require ATP, and it moves molecules from an area of high concentration to an area of low concentration. That's referred to as moving down your concentration gradient, and we'll look at that more later. Okay, so now you can watch this video, and this video is also available on Blackboard under the videos section, but we'll play it for you now. Oops. Here we go. Okay, guys, so the videos are really, really good, and I do recommend that you watch them repeatedly and kind of go through the steps with the narrator until they become very, very familiar, and do the same with the pictures in the book of the neuromuscular junction. So we're going to end this video here, and we'll go on to step 2 in the next video.